So I want to talk about the power of periodic tables. Sometimes the big discovery is a particular particle that you're studying, a gene. No, I'll just gesticulate wildly. That's okay. But sometimes the big discovery is the big picture. And that's really what we'll talk about today. Also, a theme that runs through is the speed with which science can move. And even when you're an optimist, how much faster science can move than you expect. So this will be a play in the 3 acts. Act 1, next slide. Oh, is that a clicker? Oh, if that's a clicker, I'll take it. I thought it was a pointer. You know, I'm still getting adjusted here. Super. I thought that was the point. There we go. Does that work? It does. So Act 1, the big idea. Biology is information. That took about 80 years to work out. From the early part of the 20th century, when biochemistry and genetics had nothing to do with each other. Biochemistry was purifying things in a test tube. Genetics was studying the basis of hereditary the transmission of hereditary information. And they come together in mid-century with a recognition that there was a molecule in a test tube that carried out the transmission of hereditary information, DNA. Within about a decade, the basic understanding of how DNA was turned into RNA, was turned into protein according to a simple genetic code was in hand. But of course, nobody could actually read a single letter of DNA. This was an abstract understanding, not a practical one. But within another decade, recombinant DNA had come along the scene. I show Paul Berg, a member of the Academy, who's passed away, and I'll give some memorial remarks about him later. And the ability to clone and to sequence genes came upon us. So that's 80 years it took roughly to get to that point. But all of that was retail, so to speak, gene by gene, hand-to-hand combat with an insulin gene, a growth factor gene. A new generation arose that said, no, we want the big picture. We need to get the entire human genome. And that was, of course, insane, because at the current rate of sequencing, that would have been about 200 years of work, not an attractive grant proposal, not an attractive career proposal for young scientists. But instead, the idea was, let's try to do it in 15 years. Truth is, nobody knew how, but it was a good goal. And the rough calculation, at least, that the US Congress was told would cost $300 billion. In fact, if you were careful about the arithmetic back then, 30 billion would have been a better estimate. But it actually happened. It was a remarkable project. It took about 13 years to get done. 3 billion letters of DNA were read out at $3 billion through an international collaboration. And people full well understood that we really had to choose whether we sequenced the human or the mouse, because there was no way we could do both. It was just too expensive to think about doing both of those. So finally in 2003, 2004, people could take a breath. And of course, scientists were unsatisfied because we had 3 billion letters, but we didn't know what they meant. What we really wanted was not 3 billion letters, but a periodic table. A periodic table that showed what the atoms were, what their isotopes were, what their relationships were, the rows and the columns and all the things that come from that. We had to figure out what that meant for biology, but it was a periodic table of biology. We've had about 20 years since then, and I just want to report briefly on where we're at and where it's taking us with regard to genomic medicine. So, Act 2 is the big picture and trying to get this periodic table. One sequence, one genome. Well, what did we really want? Roughly this. We wanted not just one sequence of one genome, we wanted to read millions of genomes. We wanted to know all of the functional elements encoded in those genomes. We wanted to know all the genetic variants in the population between any two people. And we wanted to know how those variants drove inherited diseases, why some people were taller or shorter or more predisposed to diabetes. We also wanted to know all the mutations that gave rise to cancers, the somatic changes that occur in the cells of your body that give rise to cancer, so all cancer mutations. We also wanted to know all the gene expression, what genes were turned on and off in every cell type of the body. And finally, of course, we wanted to know how it wasn't enough to observe, we have to perturb. We had to be able to change DNA sequences at will. And we had to be able to change the expression of genes, the RNA of genes, at will. And with all that information, we want, and this is a work still in progress, to know all the programs that cells run. The final periodic table, so to speak, is the periodic table of all cellular programs. So this was roughly an understanding, at least amongst some biologists in the early part of this century. And what has happened over the past 20 years? Well, I'll touch lightly on them. Reading millions of genomes. I said before it was $3 billion to sequence a genome. But once we started, there was a market, there was interest, there was a recognition we wanted more genomes, and technologists developed newer and better ways to sequence. And today, instead of $3 billion per human genome, I can get it for you for $300. That's a pretty good deal. I can even see a clear path to $30 per genome. I don't know that we get to $3 a genome, but then again, it costs more than that to draw the blood, so it's not clear that that's rate limiting at that point. And more than 1 million humans have been sequenced to date. More than 1 million, and it will soon become routine standard of care in the practice of medicine to have a genome sequence in the medical record, at least if a patient wants to have that. And many, many, many other species, hundreds and hundreds of mammals, at least 1/3 of 1 million microbes. I'm sure that statistic is out of date already. But we have the ability to read genomes, and we can read longer reads, shorter reads, all sorts of variations. And it's not stopping. Now, what about all of the functional elements? What do I mean by all the functional elements? Well, I mean every gene that encodes a protein coding genes. But then we came to learn that many genes encode an RNA, but it never gets translated into a protein. Those are non-coding genes. We want to know all the non-coding genes and what in the world they do if they don't make proteins. We wanted to know all the regulatory sequences where a gene is transcribed from and all the other sequences that contribute to that gene being turned off or on in a particular cell type. And we wanted to know all the proteins that bound the DNA at each location in order to accomplish those functions. There's been remarkable progress toward that from many directions coming together. RNA sequencing of lots of stuff. Take a hunk of tissue, sequence all the RNAs, begin to meet all the RNAs, and begin to sort them out into coding and non-coding and map them back onto the DNA. And then, of course, evolution has been incredibly powerful. If things matter, they tend to be conserved. So sequencing mouse and dog and rat, and by now 200 other mammals, has allowed us to spot things that evolution has lovingly preserved, even though we didn't know what they did. Epigenomic maps, being able to map the locations of all of those proteins, their modifications on the DNA by using antibodies to grab them, pull them down, sequence them. And then the genome folds up in an amazing sort of fractal origami. And methods were developed to be able to read out the folding patterns. So we never know all. When I write all, I always mean almost all. There's always a little bit more to discover, but we kind of know all these things now. Now, we wanted to know all the variation in the human population. How do you do it? Take 2 people, compare their genomes. That's pretty good. Then take 100, then take 1,000, then take 200,000. And that's been going on. And we knew at the beginning that humans differ by about 1 letter in 1,000, but now we have a nearly complete catalog. Well, I mean by that, anything that occurs at a frequency greater than 1 in 10,000 is already in the database. We know that already. We know the local correlation structure, the haplotypes, so to speak, of the genome. We can carry out massive scale genotyping, genotyping millions of people very inexpensively. We even know the variants in Neanderthals, and we can learn a lot by that comparison and about other populations, and we can read population migration and mixture, the history of selection on different genes and variants in the genome for different traits like the ability to drink milk as an adult, or functional constraints on which proteins will allow you to change them and which won't. So for all of these reasons, we wanted that information, but there was a much deeper reason. And that deeper reason was the ability to map the causes of diseases. So initially, it was rare genetic diseases, cystic fibrosis and Huntington's caused by a rare mutation. And the general paradigm was disease was due to rare mutations that interfered with a protein itself. But that didn't work very well for common diseases like diabetes and Huntington's, and like diabetes and Alzheimer's and other things like that. And what became clear that common diseases were due to common genetic variants. We needed a whole new way to do it, being able to look at the millions of common genetic variants and attribute them to different diseases. But it opened up the door to things like schizophrenia, where we could observe 300 different loci that contribute to schizophrenia, involving things like synaptic pruning and other processes, pointing us to how things worked. Cardiovascular disease, many new genes, and a tail of hundreds more that allow us to compute polygenic risk scores. There's 3% of the population that you can tell from birth have a 10-fold higher risk of cardiovascular disease. And a disease like sickle cell, a single gene, simple disease, turns out to have a very important modifier gene that is a repressor of another gene, which, if you de-repressed it, would cure sickle cell. Trust me, it's some Rube Goldberg machine, but it works very nicely there, and you can discover that in an unbiased fashion. All right, but not just inherited disease, we also wanted this for cancer. And so some early sequencing showed us some new genes that hadn't yet been seen for cancer, but they were genes belonging to an old classical type of gene in a signaling pathway. But a bunch of people said there must be other kinds of genes we never thought of. They proposed the Cancer Genome Atlas that in a systematic way would sequence tumor cells, tumor tissues. And sure enough, everybody hated it initially because they said, we know all the cancer genes, at least we know what families they belong to. But when you start looking, hundreds of new cancer genes belonging to many new types emerged. And today, it is a routine thing for cancer patients, at least in the United States, to have their tumors sequenced and have that information used to guide therapy. Now, all of this information that we're talking about was done on tissues, bulk tissues, a blood sample, a piece of muscle. But of course, that's not the action in biology. The action is at the level of single cells. And so people wanted to do the same thing, all of this same thing at the level of single individual cells. And that would mean doing the biochemistry on individual cells in droplets at high throughput, barcoding them with DNA barcodes, and reading out the information you want from every cell. So that happened. 10 years ago, that was developed. And there's a paper in Nature that reported 18 single cells. Today, we have more than 100 million single cells that have been read out this way. And it allows us to take every cell's gene expression, embed it as a point in 20,000 dimensional space for the 20,000 genes, and cluster them in this high dimensional space and find cell types. And whereas the textbook said there were several hundred cell types, we now know that the brain alone has 5,000 distinct cell types. And it's come together as a project called a Human Cell Atlas. And then, of course, it's not enough to be able to observe. We want to perturb. We want to perturb any DNA sequence. We want to perturb the RNA expression anywhere. And so having tools to do that, first RNAs to interfere, then proteins to interfere, and then the super duper combination of CRISPR, which is a combination of an RNA and a protein together that can target to any particular site in the genome and bring with it any protein you want, to cut the DNA, to activate a gene, to inactivate a gene, to insert something there. And it isn't just useful for making a change one at a time, but you can take a library of tens of thousands of these agents and combine them together. And with single cell and CRISPR, you can read out all their effects in parallel. And so we can do these kinds of studies of thousands of perturbations on hundreds of thousands of genes. So where are we today? We're today at the point where we have all of this information, and I would say the big open question in this work is, can we build a comprehensive catalog of cellular programs, all of the programs, just like all of these other periodic tables? I'm fond of that formulation because it has a nice acronym that the previous user is no longer using. And so we have hundreds of millions of cells, 20,000 gene readouts, tens of thousands of perturbations, many time points. How well can we reconstruct this? So that's where we're at 20 years after the completion of the Human Genome Project. We have a lot of these periodic tables, much faster than any of us would have dared to imagine. But Act 3, my last slide, is the big payoff. The big payoff is, what are we getting out of this for genomic medicine? Well, in medicine in the early 20th century, we largely relied on accidents. Chewing willow bark reduced your pain. We discover aspirin. It was accidents. It was throwing random chemicals at things. What we have from all these periodic tables now is a pretty good paradigm for finding valid disease targets, specific proteins, RNAs, DNA sequences, or cellular programs that, if you can modulate them in a specific cell type, will safely reverse a disease course. That's an amazingly powerful thing, is you reliably know that if only you could do this, it would really have a major effect on the disease. We also are seeing the other part of the equation, programmable therapeutics. That is, rationally predictable, designable therapeutics that can accomplish a specific goal and then can be altered to accomplish a related goal, and ways to deliver them to any given cell type. The pieces fall together, and I give you 4 examples. Casgevy, a CRISPR therapy for sickle cell disease. Sickle cell disease is caused by a problem in adult hemoglobin. If we could only turn on fetal hemoglobin, that would solve the problem. We can't turn it on because another gene represses it. And so the CRISPR goes after a regulatory sequence that shuts that gene off, which releases the fetal hemoglobin, which cures the disease. That is a rational, predictable, programmable thing. We have antibody-drug conjugates. If I know a cancer cell has a particular protein on its surface, I can couple an antibody to that, to a poison, and it can kill that cell. And there are stunning examples. The most beautiful is one in breast cancer today, of combining an antibody and a drug to do this. And this will become a programmable therapeutic. mRNA vaccines we're all familiar with. You have a particular target. In 2 weeks, you can design an mRNA vaccine if you know enough. But it's not hard to design that therapeutic and to program it to go to the right cells, macrophages. And then there are things like Ozempic, which are broadly known in the world today, which is just a plain old ordinary natural hormone that works in the human body, except it normally only has a half-life of 2 minutes. But we know how to tweak it so it has a half-life of a week, and then we have a supraphysiological effect. And this has given rise to a therapy for diabetes and obesity and many other things. So anyway, that's it. That's my brief summary of what has gone on for the past 80 years, this past 20 years, and I'll stop and hopefully we'll have time for questions.
Thank you very much, Eric.